Thank you everyone for coming. It's so nice to see everyone here today, and especially when the weather in Atlanta is such a nice weather during this time of the year, with the exception is if you have pollen allergies and it's miserable for you. So I'm based here in Atlanta, so I don't have to drive so much to get here, but for those of you who are traveling here from out of state or driving a long distance, I want to say thank you. I'm honored to be here today and to be amongst the patients and the family, as well as clinicians, researchers, in this very unique setting. I encountered my first myasthenia gravis patients when I was in my training in the 1990s. So for those of you who are good at math, please try not to calculate my age. This patient was 12 years old, and she was in the middle school. She first came to me because her voice was very soft. Then she progressed to having difficulty in swallowing, and subsequently she has generalized weakness and required intubation, have difficulty in breathing as well. So over the course of her condition, she has multiple myasthenic crises, and most of them are fairly severe. As the year goes by, with multiple myasthenia gravis crises, she underwent tons of plasma exchange, IVIG, steroid poses, in addition to Mestinon. Now, after I have treated her, there are many patients who come after her with similar stories with refractory myasthenia gravis. Now, in 1990s, there was only one approved treatment for myasthenia gravis, and that was Mestinon. Not only until 2017, a second medication was approved to treat myasthenia gravis. And this is in part due largely to you, patients, families, patient advocacy groups, as well as clinicians and researchers. Clinical research has brought a tremendous effort, motivation, and passion, and purely dedication to finding the right therapy. And as you can see, I'm very honored to be amongst our panelists, and they are from a number of pharmaceutical companies with a primary focus to do research and to bring appropriate therapy to myasthenia gravis patients.
Great. Hi, everyone. I'm Angela Pontius. I'm a Director of Clinical Operations and Patient Advocacy at Pharma. And thank you so much to the MGFA for holding such a great meeting and for inviting us here to speak to you about our clinical trials. Today I'm going to speak to you about zilucoplan for MG. These are just forward-looking statements, blah, blah, blah. All right. So before I start talking to you about zilucoplan, I wanted to introduce to you the company behind the drug. So Pharma is a small biotech company located in Cambridge, Massachusetts, and we're a team of about 80 people, and our mission is to lead the field of complement biology and bring innovative and accessible therapies to patients with rare diseases like MG. And this is just a photo of our team from this year's Rare Disease Day. So, excuse me. So I know you've heard a lot about the complement system. I won't go into too much detail because I know you're probably inundated with the information, but zilucoplan inhibits the complement system. And just a quick overview, so the complement system is part of your immune system. It has evolved to help your body fight infections. And in MG, which is an autoimmune disease, the antibodies inappropriately activate the complement in your neuromuscular junction, which is that area between the nerve cell and the muscle cell. So that causes tissue damage, and that damage makes it hard for your muscles to receive signals from your nerve cells. Like we've seen with the current approved therapy, Soliris, complement inhibition has proven benefits in patients with MG. Zilucoplan is different. Although it's a complement inhibitor, it's a small synthetic peptide. So what does that mean? It means that our drug can be dosed in a small volume, in about 0.5 milliliters. So our drug is provided in a pre-filled syringe with a 29-gauge needle. And that means it's about similar to an insulin injection. And so patients can dose their drug at home in a 5 to 8 second quick push and go about their day. So we just finished our phase 2 study, which we're really excited about. We enrolled patients with generalized myasthenia who are AChR positive, and we required patients who had a certain quantitative myasthenia gravis score. So I know you've heard a lot about that at this meeting, but for those of you who aren't familiar with it, the QMG score is a 13-item test where we measure strength. A score of 0 is really good, it's the best score you can have, and a score of 39 is the worst. And then we also allowed patients to be on stable standard of care therapies, and that includes corticosteroids or immunosuppressants. We did not require patients to be refractory, so patients didn't have to have failed multiple prior therapies. This image below shows you what our study design was like. Patients came in for a screening visit, and then at that screening visit, they consented to the study and did a few tests to make sure that they were eligible for the study. When we confirmed that they were eligible, they were then enrolled and randomized into 3 different treatment arms. So they could receive either high-dose zilucoplan, low-dose zilucoplan, or placebo. After dosing for 12 weeks at home, they were then eligible for a long-term extension. And in that extension, every patient received active drug. Our endpoints, or our main goal for the study, was to look at what was the change in that QMG score from day 1, before they started taking drug, to the end of the study. And we also looked at that patient-reported scale, the ADL, the Activities of Daily Living score, which tells us how do patients feel that their MG is affecting their daily activities. We enrolled 44 patients. So for us, that was a huge accomplishment because our target was to enroll 36 patients, and we over-enrolled. And we enrolled early. We enrolled 3 months in advance of timelines. And in our clinical trials world, that's amazing. That means that patients are interested, doctors are interested in our drug. So we're really excited and encouraged about that. So we couldn't have done that, obviously, without our amazing study sites, and their locations are shown here on the map. And I know that a lot of you are from the Midwest and West Coast and thinking, like, where are my sites? And I can let you know that in phase 3, we'll definitely have more sites in the rest of the country and around the world. So I know a lot of you have seen this slide before. You saw it in Dr. Pulley's presentation, so I won't go through it too much. But what I wanted to point out is that we had patients who did take other therapies before, but we also had patients who had never received prior therapies before going on our study. And the other thing I wanted to point out is that our baseline QMG score, which if you remember from my previous slide, is a score between 0 to 39, our average QMG score before starting our study drug was 18 or 19. So that's moderate to severe. This slide shows our study results, and I know it's a lot to take in, but basically what you'll see here is orange represents zilucoplan and the gray represents placebo. And on the left, you'll see the change in the QMG score. So even in the first week, you see that quick drop, meaning that patients' QMG was improving right away, even after 1 week of study drug. Then at the end of the 12 weeks, the average improvement was 6 points in their QMG score. And we saw the same thing in the ADL. We saw a big improvement at the end of an average of 3.4 points. Not only did we see that improvement, but we also saw that patients who were on the high dose of zilucoplan didn't need IVIG or Plex. They didn't need to be rescued. So that's really encouraging information. And we had no serious adverse events that were related to study drug. And study drug was well tolerated, and we had no other major issues. All 44 patients who enrolled in the study completed the study, and 42 out of those 44 patients decided to enter the long-term extension. So when we were asked to join this panel, we were asked, can you share what went well or what works well in clinical trials? I can speak for us, for RAW, and say that what worked well for us was that we knew that multiple people were part of the success of our study. So for raw pharma, we designed a drug that's easy to use, and we also allowed for patients to take standard of care medicine while they were on our study. We provided clear and comprehensive patient-facing documents so that patients understood what they were getting into when they joined our trial. And we also supported patient advocacy group events like this one. Of course, we had study sites who enrolled patients, provided us feedback on what was going well, what could be improved in the future, and they helped answer all of our patient questions while patients were on the study. And of course, we couldn't have a clinical trial without patients, so we're really grateful for all the patients who enrolled in our study, who read the ICF, who came to their study visits and took their study drug as directed, and reported any side effects that they had. And we're also grateful for patient advocacy groups like the MGFA, who share information about clinical trials like ours with patients like you, and also host meetings like this one, where we have a forum to discuss those trials and our data. So thank you. Sorry, I have a little bit more. Just quickly, so what's next for zilucoplan? So we just finished our phase 2 study, and we're looking into the future, and we're going to be starting our phase 3 study in the second half of this year, so keep an eye out for that one. And then what we see further into the future, and hopefully in the near future, is providing zilucoplan to a broader group of patients, just like the ones that we saw in our phase 2 trial. And with this photo, or this photo board, I should say, I just want to thank everyone who has contributed to the success of zilucoplan so far, and we're really excited about the future. Thank you.
Thank you all. I'm going to talk about 2 studies, one that's completed, and they're specifically an oral medication for MUSC myasthenia gravis, because you probably have learned earlier today that in myasthenia, not all antibodies are the same. Some bind complement in acetylcholine receptor, but in the MUSC population, they do not involve complement activation. MUSC is a subset of myasthenia, so even more rare, about approximately 8% of myasthenic patients will have the MUSC antibody versus acetylcholine receptor. And I hope everyone here knows which antibody type you have. And if not, I suggest that you talk to your physician and make sure you know whether you're acetylcholine receptor or MUSC type. So in the first study, which has been completed and published in December, we had a screening and a run-in period. So everybody starts on drug and is titrated on the oral medication to an effective dose. Those patients who demonstrate improvement are able to go to the randomization phase. Now, when you're dealing in rare diseases, you sometimes have to get very creative about study design. And these are some of the things as sponsors that we face. Because you're working with such a small number of patients, how do you demonstrate the concept in a relatively reasonable time frame with a small number of patients? Well, you may use a design like this, where actually the same patient, if randomized to either arm, is first blindly either on drug, then switches to placebo, then switches back to drug, or the opposite sequence. Okay? So this is called a switchback crossover design. Why do we do something like this? Okay, we had 10 patients. But look it, I get to use each patient and evaluate you 3 different times. And by doing that, it's like getting data on 30 patients, but by only using 10. This study, as a pilot, proof of concept, demonstrated clinically and statistically significant improvement in the activities of daily living score for patients with MUSC, myasthenia gravis. That led us into a larger pivotal study, as the FDA calls it, a phase 3, or kind of the bigger exposure for a period of time that is required in order to try and get a drug approved. The study diagram shown here is currently ongoing. And again, as I mentioned, it's a phase 3 study in the oral medication amifampridine phosphate, again, strictly in patients with MUSC, myasthenia gravis. Same basic design, but this one, we have just done a more traditional parallel group where the patient is either randomized to stay on the drug or to go to placebo. This is a preferred design when you're working with the FDA in terms of drug development. This study is currently recruiting 60 MUSC myasthenia gravis patients and a sample of 10 acetylcholine receptor patients just to explore what might happen in that group. One of the things that we can do also to try to ensure that when we put patients into a clinical trial, that whatever results we have, should they be positive, will lead to the potential for a new alternative therapy for you all, is to go into an FDA special protocol assessment. So, for example, with this protocol, we met with FDA ahead of time. We showed them the study, the design, the endpoints of MG activities of daily living, and myasthenia gravis, the quantitative QMG scale, and said, if this study, the way it is set up, should show positive results, would this be sufficient for you to consider, not to approve a drug, but to consider reviewing the drug for approval? And they said yes, and so we embarked upon this trial. One of the unique things that we were also able to do, and again, it's about talking to your physicians when enrolling in a trial and understanding the mechanism of action of the drug that you are to be treated with, is that we tried to minimize the number of days on placebo. So we understand that if we improve your condition, you then don't want to necessarily have to be on a placebo for an extended period of time. And so we worked hard to try to keep that to the shortest period that may show a difference, but still be considerate of the patient and the fact that on placebo, you may get worse. One of the things we were asked to comment or to review was challenges. So from a sponsor's perspective, what do we face in terms of challenges of setting up rare disease clinical trials? Well, one, it's to find the appropriate patients. You know, where are they? It's a rare disease. How do you find them? How do you identify them? And the patients tend to be scattered, right, throughout the country, with maybe a few here and there. And so, you know, they're going to have to travel. We can't put sites in every city. So you have to consider that and build that in. Also, large academic research centers where patients with rare diseases tend to concentrate are unfortunately still slow to get new studies started. The process, getting through their institutional review board approvals, the contracts department, the budget departments, getting it set up can take still many months. And over the decades that I've been doing this, I would like to tell you that that has improved. Unfortunately, I have not really seen significant improvements. So it tends to slow the startup and the research, which potentially brings you new therapies. Sites may only contribute a few patients, but all the startup activities that we have to do, going through all the process, educating, site initiation, shipping drug, pharmacies, is the same whether they contribute 1 patient or 10 patients. The enrollment may be sporadic, right? So there's not a steady stream of rare disease patients who are coming in 2, 3 a month. And what we find is that it may be gaps between patients. And the site coordinators, the investigators forget. They're doing several other studies. And they forget the procedures. They don't remember quite the protocol. So for a sponsor, we have to go in and actually keep retraining them and re-educating them when a new patient comes along. And then the other thing is high turnover of research coordinators at clinical study sites. You know, I think research coordinators are the ones who really do the work at a study site. And most of them have multiple trials and are just unfortunately not paid that well. And there's a constant turnover at a lot of sites, which again means more retraining. You know, information isn't passed on to the next one, and you kind of keep starting again. And these are the challenges that we face in order to get high-quality research available to you all. Overcoming those challenges, sponsors, especially in rare diseases, have to highly value and respect every patient who is willing to participate in a trial and make that experience for you all as easy, as simple, and as painless as possible. How to find the patients. Today, we utilize a lot of social media to help find patients. Because patients today are quite educated. You're out there. You're searching for answers, for information, for what's new, for how to better treat your disease. And we found that social media is very helpful in terms of connecting with patients and then seeing if they are appropriate and interested in a clinical trial that has the potential to benefit them. Provide travel support, okay, through a rare disease organization or other third party. We know we're going to have to transport you to a site, right? Because we just can't open sites everywhere. So again, do we cover those costs? Make that simple. If you need an overnight stay between 2 visits, make that possible. It shouldn't cost you anything to participate in a clinical trial. We also now have started to utilize trained, independent clinical research centers to handle patients in an expeditious manner. What that means is not so much the typical university, but actually a site that is geared to only doing clinical research and can get you in according to your schedule. Finally, as patients who are considering clinical trials, some of the things I would encourage you to ask of sites and of your physicians is, will the trial sponsor meet your needs in order to facilitate participation with travel assistance or if you require a caregiver? Yes, while there are concerns about taking an experimental medication, remember, we are in a highly regulated environment. We have to report, record everything. You will get probably some of the best care and attention you will ever receive in a clinical trial. And personally, as a patient, I would be concerned when I'm given therapies that have not been studied specifically for my disease, and what are the long-term effects. And then finally, your participation in a clinical trial really is what will make new potential therapies available for everyone who can benefit with your condition. Thank you.
Okay, so I'm going to talk a little bit about our program that brought Soliris to market. As was previously pointed out, it's the first new therapy in however many decades, which was approved in 2017. So this is a high-level overview of our clinical program. It took a pretty traditional path, starting with a phase 2 study of 14 patients in a crossover design similar to what you saw in the other phase 2 studies that were presented by the other presenters. We then went into a single pivotal phase 3 program called the REGAIN study, followed by an open label extension period where patients, regardless of whether they've been assigned to placebo or eculizumab or Soliris, in the main study were allowed to get open label drug. So I'm going to focus a little bit on our pivotal study, REGAIN. This is a diagram of how the study was designed. Started with a screening period. Very importantly, patients need to be immunized against meningococcal infection because the one risk that we think most about with complement inhibition is susceptibility to meningococcal infection. So all patients were immunized at least 2 weeks before receiving drug. They were then randomized 1-to-1 to receive either eculizumab or placebo for a period of 26 weeks. And as I mentioned, all patients at the completion of the study were given the opportunity to roll into the open label study where they all received eculizumab. Our inclusion criteria, importantly, we focused only on anti-AChR antibodies. This is because we know that the anti-AChR antibodies fix complement and thus feed into our mechanism of action. Catalyst is doing work with MUSC, which, as was pointed out, does not fix complement. So we would not have expected our drug to be efficacious in that population. We had a pretty broad range of severities with MGFA clinical classification between 2 and 4, and MG-ADL score greater than 6. And finally, we required a certain level of refractoriness. So all patients had to have tried at least 2 therapies in the past and failed. The primary endpoint was change from baseline in MG-ADL, which, as many of you know, is a patient-reported outcome. It's basically 8 questions that evaluate your state of well-being over the past 7 days. Secondary endpoints included other functional measures, including QMG, MGC, and MGQOL-15. We also did responder analyses on the MG-ADL and QMG, assessing the percentage of patients who had clinically significant responses on those measures. So here's where our study gets interesting and a little bit different than most of the research certainly I've done and that I've seen in the field. The way we analyzed statistically our primary endpoint was using a technique called the worst rank analysis. And the rationale behind this technique is that in the course of a trial, if a patient has a clinical deterioration, ethically, we want to do the best for the patient and give that patient rescue therapy, knowing that giving that rescue therapy is potentially going to confound our endpoint. If they get better, we don't know, was it because of our drug or was it because of the rescue therapy? So the worst rank analysis allows us to account for those patients, get useful information from those patients while doing the right thing for them and making sure they have access to appropriate patient care. The way this is done is that we rank patients in order of outcome from best to worst, and we have different categories of rankings, which I'll go over in a little bit more detail in this slide here. So the best patients, of course, are the ones who get better on the endpoint, who don't have clinical deteriorations, don't have any other significant events that cause them to drop out of the study. Those are ranked from best to worst by the magnitude of improvement of the endpoint, in this case for the primary endpoint, MG-ADL. In the poor outcomes category, we have several subcategories. The worst is death. That's someone who dies is ranked absolutely bottom. MG crisis is the second worst. And then in the intermediate area, we have patients who either required rescue therapy or who discontinued the study for any reason. And as you'll see, that became a very key issue in the analysis of our trial. So actually, let me just go back. Is there a way to, can you help me go back a slide? Thank you. I should give the results of the trial. We went through it. So, when you finish a trial, it's weeks and hours and late nights cleaning the data, locking the database, and then everybody sits on the edge of their seat when they get the results. So what we found was that the mean rank for eculizumab was 56.6 compared to 68.3, which showed that at least numerically eculizumab did better than placebo. However, the p-value, which is an important measure of statistical significance, was 0.0698. Now, traditionally, anything less than 0.05 is considered statistically significant. So by our own pre-specified standards, we missed our primary endpoint. And so you can imagine the reaction that caused within the company and within patient groups as they learned the results of our study. So many people thought we should give up. The FDA initially was not interested in hearing about our study because they said, you failed your primary endpoint. You don't have a filing. But we were very stubborn. We were not ready to give up because we really believed that this drug worked. So we did an analysis of what caused us to fail our primary endpoint. And one thing we did is we went back at the history who had a spa? You had a spa. So did you have the spa? You had the spa, yes. So we also went through a process where we had, prior to doing the study, we had consulted with the FDA to get their advice. And one thing we wrangled with them a bit was on exactly how we would define the worst rank analysis. They were very, very conservative, and what they said in the end was, we want you to take every patient who discontinues, no matter how they were doing when they discontinued, no matter why they discontinued, and put them low in the worst rank analysis. Just assume, to be conservative, that they did poorly. And that's a difference we made between version 2 of our statistical plan and version 3. In version 2, we were a little bit more nuanced. We looked at patients who discontinued, and we asked, why did they discontinue? If they discontinued because they were getting worse, or if they met any criteria for deterioration, we said, yes, this makes sense. This patient is doing poorly, they go low. But if they discontinued for another reason, for example, if they moved away from the study site, or they just got sick of doing the study because they felt better and they were going out more, so they were doing better by MG-ADL at the time of discontinuation, we took that number, that score at the end of their participation, and used that to rank them higher in the worst rank analysis. And I'll show you the result was that we had, by chance, an imbalance in the number of patients who discontinued. 4 patients in the eculizumab group discontinued due to adverse events. None in the placebo group discontinued. And you'll see, digging down into why patients discontinued, one had a Moraxella bacteremia infection, but they were doing better at the time of discontinuation with a 3-point improvement in MG-ADL. One had a bowel perforation, which was felt by the investigator likely to be due to cell sepsis, nothing to do with our drug. They were actually doing very well on our drug. They had 7-point improvement in MG-ADL at the time of discontinuation. Another patient had stage 4 prostate cancer. This is, to get to stage 4 takes years and years. Clearly, this was not caused by our drug. They were doing really well. They had a 7-point improvement at the time of discontinuation. And finally, the 4th one was a genuine failure. This person had a myasthenia crisis, so we acknowledged that patient did not do well with our drug. But the other 3, we thought it doesn't really make sense to put them in the very bottom of the rank when they were doing so well on our drug, and they dropped out for reasons that were really unrelated to the drug treatment. So this is a comparison of how the ranks stack up. Looking at the left side, this is under the very conservative version 3 of our SAP, and on the right is version 2 of the SAP, or the statistical analysis plan. And you'll see in the highlighted or the grayed out regions the difference it made when we looked at with a little bit more nuance at how patients were doing and ranked them according to how they were doing. So when we reran the analysis under the second version of the SAP, we ended up with a strongly positive endpoint with a p-value of 0.014. So this by itself was not necessarily going to win over the FDA. However, in designing our study, we were very careful to look at a number of different endpoints so we get the whole picture, not to hang the entire success or failure of the trial on a single endpoint. And you'll see here on this grid, this just goes over 22 endpoints that we looked at, or 22 pre-specified analyses. And you'll see that 18 out of 22 of them had p-values that were less than 0.05. The ones that failed due to the way we defined our worst rank analysis. And when we went back and ran the worst rank analysis under what we considered to be a more rational approach, we achieved significant p-values in those patients. And this is just a look at what the data really looked like. It's a little bit hard to conceptualize, at least for me, what worst rank means. But this actually shows the change over time in MG-ADL and QMG, with the red curves being patients on placebo, blue curves patient on eculizumab. So you see a very early and substantial split with a lot more improvement in the eculizumab patients. One thing that's interesting that we see across other trials that use MG-ADL and QMG is that there is a pretty strong placebo effect. And part of this, I think, has to do with the fact that I think you had mentioned that going into a clinical trial, you get some of the best care you've ever had. So patients get a lot more attention in clinical trials, which is one reason a lot of patients like to be in clinical trials. And as a result, especially in neurology trials, we do tend to see a very strong placebo effect. But nonetheless, even with that strong placebo effect, we had a very strong statistically significant split in outcomes between patients who were treated and those who received placebo. And this is a responder analysis. Blue is the patients who were treated, red is placebo. You see at whatever threshold we set as defined as a responder, we did better with patients on drug than placebo. So finally, just to summarize, the REGAIN trial narrowly missed statistical significance on its primary endpoint, which nearly killed the program. But as we dug deeper and didn't give up, we found a good explanation for why we missed our primary endpoint, and found that with a slightly different set of rules for assigning worst ranks, we actually achieved significance on all of our endpoints. This treatment effect was consistent across an array of complementary endpoints, showing overall consistency of effect. And as a result of this overall analysis, we were able to convince all the major regulatory agencies, the US, Canada, Europe, Japan, and many other countries to approve our drug. Thank you.
Good afternoon. I'm thrilled to be here and to have the honor to talk to you today. Now, I first need to understand how this works. I got it. Do you hear me? Yeah, okay. So I'll just try to keep it short and sweet. I'll speak a bit about argenx, the company, then go to efgartigimod, the FC fragment we have, which is an FcRn inhibitor, and then we'll talk about the clinical development and our phase 3 that is running right now. So argenx, probably most of you haven't heard about argenx before this weekend. We are a global biotech company with roots in Belgium, activities in Boston and Tokyo, and we're specialized in antibodies. At argenx, we do have a mission, and it starts with both the underlying biology and patient needs. And we aspire to become a recognized leader in translating biological breakthroughs into novel treatments that truly impact patient lives. So we stay true to our mission with the design of efgartigimod, where biology played an eminent role, and we looked for a disease that would match that biology, like that key and lock principle. And since it was reducing IgGs, autoimmune diseases needing a reduction in IgGs were an important indication, Myasthenia gravis being one of them. We focus on rare diseases with efgartigimod, that FC fragment, and I'll come to it and speak about the biology in a minute. And not only rare diseases, but rare diseases with limited and inadequate treatment options. As you can see on the slide, we're present in neuromuscular diseases like myasthenia gravis and CIDP, but also in hematology and dermatology. Now, allow me to just spend a few minutes on the pathophysiology of myasthenia gravis. And as you all know here in the room, IgG antibodies are an essential part of our immune system. They protect us from diseases. They protect us from foreign invaders. Now, having said that, in autoimmune diseases, some components of our body turn against ourselves. And in MG, we do have specifically those IgG antibodies that are here in red, those Y-shaped proteins that are turned against the body. And the role that they play is in the neuromuscular junction. And you see that neuromuscular junction with the nerve on the upper side and then the muscle. And the nerve emits an electric signal that is then transformed into a chemical signal with acetylcholine that binds to the receptors and then conveys the signal to the muscle to contract. Now, exactly that is impaired in myasthenia gravis, as you probably all know. And those antibodies block, first of all, the acetylcholine from binding, but second, they also reduce the number of receptors in that synapse. And third, they recruit complement, which is another part of the immune system, and we heard about it just a few minutes ago, that do destroy the surface of the muscle. Now, on this slide, you have the reason why we think that targeting the FcRn pathway might be a solution to that, to exactly target the underlying cause in myasthenia gravis. What you have here is in pink, my son would call it pink, I'm not sure this is pink, a cell with that recycling mechanism of those Y-shaped proteins, which are IgG's antibodies. And this recycling mechanism is something that the body has developed over evolution to keep those IgGs as long as possible in the body to protect us from foreign organisms. Now, this is only possible with that, and I'm not sure we do have a pointer. Yes, we do. But then I should go over the heads. Well, I'm not going to do that because I don't want to hurt my competitors here. So having said that, we do have that recycling mechanism, and this is only possible thanks to that FcRn receptor that looks like a bit like headphones in blue. And that attaches the antibody, and then the antibody is engulfed into the cell and recycled back into the bloodstream. Now, if we think of MG, where the problem really lies in those IgG antibodies, we could actually reduce their numbers and then get rid of the problem. And this is exactly what we're doing. Here you have a new component into the drawing, that green little thing that is sitting on the FcRn receptor. That is efgartigimod, that Fc fragment. And Fc fragment, it's called because it's just one little part of an antibody. So it's just the tail that sits on that FcRn receptor. And by sitting on the FcRn receptor, it blocks it. And so the IgG antibody cannot dock to that FcRn receptor anymore. So instead of being recycled, it's actually being eliminated into the garbage disposal of the cell, which is a natural way to get rid of antibodies, by the way. So then you see that nothing is recycled anymore, and a cause of that, or a consequence of that is the direct reduction of IgGs. So theoretical understanding and a firm grasp on biology is a very important thing. However, you need clinical data to do your job. And so what we did is we completed a phase 1 healthy volunteer trial and then embarked on the phase 2 that you see here in front of you that we completed last year. And by the way, before I go into further details, this phase 2 was one of the fastest recruiting trials in MG ever. And I would like to express my gratitude to all the patients who enrolled into that trial because they helped understand the FcRn, also our Fc fragment, but most and foremost, they helped us understand how myasthenia gravis really works. So a big thank you to those patients. Now, if we go back to the design, we see that there was a screening period and there was a treatment period. And that treatment period consisted of 4 doses, weekly doses, which were intravenous doses of efgartigimod, of that FC fragment, that were given intravenously, and then the patients were followed up. The patients were observed for safety and efficacy. Then we put all the data together and went to see the FDA, and together with them, we then decided to embark onto a phase 3 trial, which is running. But before we go there, we actually asked the MGFA, we asked the clinical experts, and we asked you, the patients, what exactly would be a good trial. And I think what is important is that we also heard of that frustration of the patients who are suffering from MUSC or LRP4 or seronegative myasthenia gravis. Those patients are usually being excluded of the big trials. And this is why we actually changed that, and patients in that phase 3 are eligible. So MUSC patients, LRP4, and seronegative patients, also acetylcholine receptor positive patients, are eligible for that trial. Another thing was that patients wanted to have a therapy that targeted the underlying cause of MG. And I think that I demonstrated to you that we think that we really have a mechanism of action that actually targets the underlying cause of MG by reducing those disease-driving IgGs. On the top of that, they wanted to remain on their standard of care, on their current medication. So that's why we're dosing efgartigimod on the top of their standard of care or of placebo. One of the most important points, for me at least, as a chief medical officer, is to recognize that all the patients are different. And all those patients do have different needs. And that's why we have decided to tailor the approach, to tailor the treatment. And that means, in other words, that treatments will be scheduled and given based on physician's evaluation of the symptoms. Having said that, this is the phase 3 trial that will aim at enrolling 150 patients. It will run in North America, Europe, and Japan. It's a randomized, double-blind, placebo-controlled, multi-center trial, as usual for a phase 3. As I was saying before, it will enroll acetylcholine receptor positive, MUSC, LRP4, and seronegative patients. And most importantly, after that, or those 26 weeks of treatment, there will be a rollover phase into an open label extension. So whether you were on efgartigimod or on placebo, you will have the possibility to be exposed to efgartigimod in the open label extension trial. The first patient was dosed in September 2018, and the study is ongoing. Just a few slides before I conclude this presentation. What to expect of such a trial? Well, first of all, you'll have a screening period of up to 2 weeks, what is actually normal in such a trial, where you'll be tested on eligibility. Then there will be the informed consent, where you're encouraged to ask any question. And once you are randomized, you'll be randomized either on placebo or efgartigimod. And since it's double-blind, neither you nor the study nurse or physician or nor us will know on which treatment you are. Treatment will be given by a healthcare provider via infusion over 1 hour. The efficacy will be measured with those 2 recognized scores that we heard before, the myasthenia gravis activities of daily living, the ADL score, and the QMG, which is an assessment of the physician, while the other one is patient's assessment. And I would like to conclude this presentation with a big, big thank you to the MGFA for everything that you're doing for patients and for the clinical research in MG, but also to you, the patients, for enabling us to do that clinical research. If you have any question, please drop by at the booth. I'm assuming that my colleagues here in the front will be more than happy to answer any question you have. Thank you very much.
I don't have the height issue that the other men were having, so I don't think you'll have any problems hearing me, luckily, with the microphone. I don't have any slides. I just want to take a couple of moments. I know that we are a little short on time as well, so I'll try to be brief, but I wanted to tell you a little bit about Momenta and what we're working on. And if you have any questions, our Chief Medical Officer is also here and can certainly talk more about, elaborate a little bit more than about M281, which is Momenta's product. My colleagues here, as we pointed out, have talked a lot about it. But M281 is an anti-FcRn antibody. You've heard those already. Obviously looking to study this to impact and improve the clinical signs and symptoms of myasthenia gravis. And it blocks that FcRn-mediated IgG recycling that we've heard talked about. And it really then will reduce the antibodies that are in the system, and it's both AChR and the anti-MUSC. So we're including both of those in our phase 2 study, which is getting off the ground now, and it is for adults with generalized myasthenia gravis as well. The things that I have we've talked a lot about rare diseases. It's a very different realm. I've been doing clinical trials for a very long time, and you have to know better, you have to do better, you have to think smarter. Because every patient is really valuable in a way that maybe you don't see in some of the more bigger studies and bigger diseases. But the things that I feel that we've done really well at Momenta is that we've been a very patient-focused in our design. We've had a lot of FDA engagements. We've sought feedback from agencies outside the FDA and in other parts of the world as well to understand, what should we be looking at, what should we be doing, what's important. And our study has 5 different arms, so there's 5 different possible doses, and there is a 4 out of 5 chance that you would be on the M281 product, and so there is 1 placebo arm. But we felt like that was a beneficial way to design this trial to give you a 4 out of 5 chance to be on M281. We also have what we call an open label extension study. So anybody who participates in this phase 2 study would then have the option to continue on and extend participation, and it would be on M281. So even if you happen to have been on a placebo as part of the initial phase 2 portion of the trial, there is the opportunity to have M281 for up to a year. And, our goal has really been to always understand what is the best dose. We have to understand the dosing levels and intervals and be able to give the doctors and the patients flexibility to say what works for you. We've heard a lot about MG patients. It's no 2 cases are identical. And so being able to have an option that you can tailor to what works for you versus someone else has really been an important focus for us. So Momenta's really excited to be partnering with MGFA and all of you, and this is my first time here at this meeting, and I've really enjoyed it and enjoyed getting to meet so many patients directly. It's something that we sometimes we forget, and it's nice to not take for granted why we do what we do every day. So thank you for having me here, and if you have any questions afterwards, of course, we're happy to answer any of them. Thank you.
All right, so again, as mentioned, Christine Coquery, I'm Senior Director of Immunology at Immunovant. The first thing I'd like to do is thank the MGFA for giving us the opportunity to speak at this very important symposium and be on this panel. So thank you all so much, and thanks to all of you for being here this afternoon to learn more about what we're doing and how we're all trying to help the patients by coming up with some new therapies that hopefully you'll be able to take advantage of. So for many of you, over the past couple of days, we've had an opportunity to engage with you, to talk to you and tell you a little bit about Immunovant. And for most of you, you have probably never heard of us. I'd like to point out half the company is in this room today. So there are 6 of us, and we represent half of the company. And some of my colleagues are sitting here. Others here, I'm missing Bev, but she's out there as well. So we're all here to support all of the MGers and all of the community that's working hard to really understand more about your disease and provide you with some options for your treatment. So who are we? other than the 6 of us. We're a nimble and innovative biotech company. We have a breadth of experience across therapeutic areas. My specific experience, I've focused in immunology my entire career with an emphasis in autoimmune disease. So I'm a B-cell expert by training. So of course it's very exciting for me to be in this space and to be talking to all of you. So our mission is really to develop novel therapies that treat your symptoms, but also, and maybe something we'll get into a little bit later, help modify the course of your disease. Something that you've maybe heard in other talks, and hopefully we'll get into that a little bit today. So how do we do it? We intend to expediently develop and commercialize a unique portfolio of clinically and commercially promising assets spanning multiple therapeutic areas as well as mechanisms of action. Today we're only going to be talking about one of them, which is our investigational product RVT-1401 for the treatment of Myasthenia Gravis. So I'll tell you a little bit about the molecule because really my colleagues and my research partners have told you everything about the mechanism. So it's a fully human, novel, high-affinity monoclonal antibody. It targets FcRn. We'll talk a little bit more about that in the next slides, but the great thing is for the past couple of days, you've been inundated and heard a lot more about that receptor and how it may be important, and we may be able to target it to help treat your disease. So far we have performed a first-in-human phase 1 study in healthy volunteers. I'll show you a little bit of data from that in the next slides. And we've seen that after a single dose, we have a marked reduction in IgG. And after 3 doses of our product, we can get IgG levels down to 80% of baseline. Importantly, we're working with a fixed-dose subcutaneous injection. We believe there will be no need for IV induction. And as you heard from our speaker from Raw, this is an injection, which is very different than a sub-Q infusion. The injection takes about 10 seconds. So we're evaluating weekly administration in our upcoming MG trial. However, we're also evaluating the potential to do less frequent administrations. We know that's important for patients. The molecule itself, so just a little bit more about the molecule, it's been designed to reduce the risk of immunogenicity as well as effector functions. So the mechanism of action, I will spend almost no time belaboring the point that everybody knows that a lot of your symptoms from MG arise from autoantibodies that target different proteins of the neuromuscular junction. So a lot of emphasis on AChR, a lot of emphasis on MUSC, but we know there are so many of you that are what we call seronegative. But you've heard a lot that we don't necessarily believe there's any such thing as seronegative. We just haven't been able to find the protein that your antibodies react to. But we do believe they're all part of the pathway that involves in signaling and allowing for muscles to contract. So RVT-1401, as I said, is an anti-FcRn. You've seen similar figures from other people who have already presented, but I want to explain a little bit more about how this works. So the cell is going to take up proteins, okay? So there's a lot of mechanisms by which this happens. It takes up proteins. That includes IgG, that's normal IgG. It's also going to include our drug after administration. And what happens is when those proteins enter the cell, they're going to go into a little pocket. That little pocket is called an endosome. It's an acidified pocket. And what happens is normally IgG binds to FcRn and it gets shuttled back to the surface. And you've heard a lot of people talk about how that maintains IgG in your body. This is very important in terms of your adaptive immune response and protection against infection. However, for those of you suffering from MG, that's a problem when a lot of that IgG is pathogenic. So what our drug does, and I will again save everybody the laser pointer over here, is that our drug binds specifically to FcRn and it prevents IgG from binding to it. And so what happens is our molecule bound to FcRn gets recycled back to the surface, whereas the IgG goes into another little compartment. That compartment's called the lysosome. And in that compartment, it gets chopped up, and it can become something else when it becomes amino acids again. So I want to share with you some data. This is not data that's really been shown publicly as of this point, and we're actually going to be showing a fairly full data set from our first-in-human study at AAN, so it sounds like a lot of good presentations coming up at AAN, which is just in about 5 weeks from now. But this is looking at IgG levels in healthy volunteers. So if you look first, those little red arrows talk about the dosing strategy. So it's a once-weekly subcutaneous injection that these patients received, and you can see a fairly marked reduction in IgG, down to about 80% was our max reduction. The average is sitting at about 78%. So I want to talk a little bit about our doses. So here we're showing you 2 doses, 340 milligram and 680 milligram. This probably seems like a lot, but this is a fixed dose. So if we kind of work to translate what that means to what you've probably seen from other individuals in their data into a milligram per kilogram basis, 340, if you're a 70 kilogram person, about 155 pounds, means less than 5 milligrams per kilogram. If you translate it into 680 milligrams, and again, we're talking about a 70 kilogram or 155 pound person, then you're looking at a little less than 10 milligrams per kilogram that we're administering. So we'll get into our trial, and our phase 2A trial is just about to start. This is a randomized, double-blind, placebo-controlled study of RVT-1401 in MG patients. As most of you have seen before, our inclusion criteria are pretty consistent with almost every trial. This is just a snapshot of them, and I'm not going to go through them line by line, but we have a screening period of up to 21 days, followed by 6 weeks of dosing. We're evaluating 2 different doses of RVT-1401, and then we do have a placebo arm. So again, as you've heard from a lot of people, we're trying to ensure that MG patients, there's a greater likelihood of you getting on study drug. So in this case, 2/3 of patients will receive study drug. So patients will receive a once-weekly subcutaneous injection for 6 weeks, and then we'll continue to follow you up for an additional 12 weeks. And so our primary endpoints, again, because this is a very small study, and you've heard this, MG, you all know how rare and special you are, so we just want to try to capture as much information as we can with as few patients as possible to achieve that. So our primary in our study is actually safety, and so we're going to be looking at safety and tolerability. We're also going to be looking at PD endpoints, and a PD endpoint is a pharmacodynamic endpoint. It's a fancy science term. It just means we're going to be looking at IgG levels. So we'll be looking at total IgG, we'll be looking at IgG subclass, and very importantly, we're going to be looking at anti-AChR titers, right? And we're going to be looking for all of those to go down while you're on study drug. However, we do believe that we should expect to see potentially some clinical response, and so secondary endpoints are looking at those clinical measurements, and you've heard all about those composite scores as well as all of those patient-focused scores, quality of life scores. So we're looking at QMG, MG-ADL, the MG composite, as well as the MG-QOL, R15R, excuse me. We'll also be looking at pharmacokinetics, and so that's just the drug profile, looking to see that that drug goes up and then it comes back down. And we'll also be looking at immunogenicity, an important part of any monoclonal antibody program. So we're early on, right? We haven't even started our phase 2 trial yet. So when we were asked to be part of this panel and talk about some lessons learned, we had only a small amount to draw from. But one thing we wanted to share with you, which is something that we did before we even started writing our study protocol for our phase 2A study, is we put together a focus group. And we were very fortunate to partner with a company called MindSpot, as well as with MGFA. And what we put together was this group of MG patients, this is an online focus group, to get some feedback from them about their disease, about their experience with clinical trials, about what they'd like to see from them. And I've put some demographic information on this slide about the type of patients that we had speaking to us, and we understand it doesn't reflect all MG patients, but it did give us a snapshot, and it did give us some really important information that we used as we designed not only our trial protocol, but what were some of the special things that we were going to do to try to help patients based on their feedback. So I have this, I like this, it's a word cloud. So during the course of that focus group, which we were only allowed to listen in on and actually see the writing, we weren't actually allowed to directly engage with MG patients as part of it. We really wanted them to feel free to speak and tell us really how they felt. We got a lot of feedback on the types of symptoms that they experienced most, and this word cloud kind of shows you what some of those were. So obviously the larger the font and the bigger the size of the word there, the more it impacted those patients. So double vision, of course, being the one symptom that affected this group of individuals the most. Now, what we've all heard over the past few days is how many more symptoms there are that aren't even reflected in this word cloud, that we really do need to start to think about how do we incorporate looking at those symptoms in some of these trials. Some of those symptoms aren't reflected in any of the composite scores, in any of the parameters that we've looked at so far. So as a community, we really do need to think about that, making sure we're capturing the thing that bothers all MG patients. So something, food for thought for all of us, including our company, as we go into our next set of trials. So a couple of the key things that came out of that discussion was that it's really hard to get to the trials, okay? The trial sites are difficult to get to, and the schedule is really hard to adhere to. So in a clinical trial, you're getting great care. Everybody's talked about that, right? But you have to go in an awful lot to see your physician, to see a PA, to see a nurse as part of that trial. So we thought, what can we do to make that a little bit easier? And we took two strategies. So our first strategy was partner with a group called MRN. MRN is a home health nursing company. And what they'll do is they'll come to your home. And so we have some visits that are a component of our trial that don't require a clinical assessment. And in those cases, patients are going to have an opportunity to take advantage of that program so that the nurse comes to your home, draws your blood there, takes your vitals there. You never even have to go into the clinic. Now, if you want to go into the clinic, you can opt out of that. It's completely up to you. So it gives you the flexibility, taking advantage of that service, and simply not having to go into the clinic for every single visit in our trial. The other thing we did is we partnered with MD Group. So MD Group provides patient concierge services. We recognize, and we've seen, the nice graphic of where the clinical sites were for the raw trial. Most of our trials are in similar places. There's only so many experts in the MG field. There's only so many places that are interested in even participating in clinical trials. And so what we know is that sometimes it's hard to get to. This concierge service is going to help you with flights. They're going to help you with travel vouchers. They're going to help you with Lyft or Uber or whatever your preferred mechanism of vehicle transport is. Also with accommodations overnight, if that's something that's needed to get you to a center to be able to participate. So we are listening, and we are trying to make it a little bit easier to be part of these studies. So with that, we'll just say thank you. Really appreciate everybody being here. Really appreciate all of the feedback that we've gotten over the past few days. We really look forward to working with you moving forward. So